And the last speaker of this session, last but not least, Malu Tansey, who's going to talk about GBA1 and the immune system. And we all know Malu's been really a pioneer in looking at immune function in Parkinson's. She was doing this way before it became popular and hot. So look forward to the talk.
Thanks, Ted. Really appreciate the invitation to speak at this amazing inaugural meeting. So thank you, Ziv, and everyone else who put this program together. I'm going to give a little bit of a different talk than what you've been hearing so far. One, it's going to be more immune-related. And two, I hope to stir up some controversy. These are my disclosures. And I want to start out with this debunking the idea that the brain is immune privileged, right? We know it isn't. I think as neuroscientists, we were led astray for many years thinking that the brain was immune privileged. If anything, it's immune specialized. And so there is crosstalk between the central nervous system, the peripheral nervous system. Now we also know there's also crosstalk with the gut. And this communication that is bidirectional in many instances is critical for brain health. And so one of the key things that we need to think about as neuroscientists and neurologists interested in brain diseases is what are the functional relationships between these sites? What are the microbial-derived molecules? What are the peripheral immune cell-derived molecules that act on the brain? And how is the brain talking back to these peripheral organs? And so today I'm going to mostly focus on our work in the peripheral immune system. And I want to remind you that inflammation in the periphery is very different than inflammation in your brain. In the periphery, you have the Celsius signs that we've all heard about, and most of you that went to med school had to know them, right? So rubor, tumor, calor, and dolor, and then loss of function. And in the CNS, you do not have that. If you did, you would have extremely dangerous intracranial pressure. And so the microglia, or brain resident innate immune cells, are a lot daintier than that. They are not like the cousins in the periphery, the macrophages. And so every immune response in the brain must be graded and must be controlled, and everyone must go back in their cages. We know there's drainage of CNS antigens and lymphocytes to deep cervical lymph nodes. And we know that one of the important roles of the microglia is to phagocytose debris and dead cells. But I suspect they don't do it alone. And so we and others have proposed that they are sending signals to the periphery, to the cousins, to get some help, or perhaps the cousins just show up anyway because the microglia are not playing with a full deck. And so it's important to understand this communication. We know that the immune system is an important partner of the brain and it keeps it healthy. And we actually think of it as the first responder. When there's organ or brain dysfunction, they are the ones that are going to be sending us clues to maybe intervene. And so we focus on this conversation and how it changes with age, environmental factors, physical activity or inactivity, lifestyle choices, infections, stress. And understanding this conversation, I'm very confident, will lead us to understanding how the immune system protects or predisposes an organism or an individual to neurological conditions. And so we also focus on the gut-brain axis as a useful model in which to study this neuroimmune crosstalk. And LRRK2 and GBA have emerged as key regulators of this crosstalk. I'm not going to talk to you about this. I did last week in Crete at the LRRK2 meeting, but I'm going to focus on the GBA findings in the periphery. So this is from one of our reviews. We think of the perfect storm for Parkinson's development as a combination of aging and gene by environment interactions. And we know that one of the things that has been vastly understudied in neurological, age-acquired neurological disease is the effect of aging on the immune system. And so what we feel is that if we were to focus on how the immune system might be sending us clues in the earliest stages, and even better, the prodromal stages of these diseases, we may be able to intervene and perhaps use that information to develop better targeted therapies to slow down, delay, or arrest the progression to motor symptoms, for instance, in the case of Parkinson's. And so to tell you a little about immune system aging, because of infectious disease, there's been strong evolutionary pressure to develop strong genes that give you immune responses and strong, robust immune responses so that you can pass on your genes, get to reproductive age, and after that, nature doesn't care what happens. And because we've lived too long, the process of inflammaging, which I'll describe shortly, as well as what we call chronic antigenic load, which is repeated infections and repeated exposures, these things converge to accelerate immune aging or immunosenescence. And I do think that as people who are interested in age-acquired neurodegenerative disease like Parkinson's, we need to consider immune aging as a real critical factor. And so your Garanzi and colleagues have studied the aging of the human immune system, and it's a bit of a two-headed monster. On one side, you have this age-related immunodeficiency, and on the other, you have an age-related inflammatory syndrome. And so not to get too bogged down with the details, the outcome of these two things is that you end up with impaired innate and adaptive immune responses. And you end up with constitutive low-grade inflammation and a kind of age-acquired autoimmunity. And so if innate immune dysfunction is the root cause of chronic inflammation and the immune system either over-responding or under-responding, we need to look at how the factors that give rise to innate immune dysfunction, including aging, including environment and genetics, how those are contributing to neurodegeneration. It's really not that the immune system becomes activated after neurons die. It's not that the immune system becomes activated once protein accumulates. I would propose that proteins accumulate because the immune system stops doing its job over many years. So we know that there are inflammatory manifestations in Parkinson's, both at postmortem in the brain. We've known that for quite some time. We also know that there are antigen-specific T cells that have seen synuclein in a good proportion of Parkinson's patients. We know that there are profiles for cytokines in the biofluids, like the plasma and the CSF, are all out of whack, right? It doesn't really matter if they're low or if they're high. They're just not normal. And even a pro-inflammatory cytokine needs to do its normal function. And so what I'm going to propose to you today is that we probably need to stop looking at just the biofluids, and we need to be studying the cells in the immune system that make these cytokines. And I think to Greg's point, it's important to see whether there's innate immune dysfunction and whether there's a trait as opposed to a state, which you can capture in a biofluid, but you can capture an immune dysfunction trait much better if you plate the cells and then challenge them. So that's exactly what we have chosen to do. PD genetics implicates a role in the immune system in pathogenesis, and both LRRK2 and GBA are highly expressed in monocytes and have been implicated in immune function and inflammation. We also know that genes and risk factors are their modifiers of PD, and many of them converge on the lysosome. And so in my lab, maybe because I was never able to focus, we work on all of them. We work on LRRK2, progranulin, GPNMB, GBA, and we work not so much on a particular disease, but on what we can learn about the neuroimmune crosstalk dysregulation across diseases. Because I do think that if we understand that, we may be able to develop therapies that apply to more than one disease. So I'm not going to spend any time on GK since we had excellent talks this morning, but just to tell you that we are focused on the inflammation connection to GBA1 mutations. And just to remind you, since I will show you some LRRK2 data, that LRRK2 and GBA1 are two genes that are very important to familial Parkinson's. But the dysregulation of these two proteins is also present in sporadic cases. So why look at LRRK2 and GBA? Sometimes in conjunction. Today I'll primarily focus on the GBA1 mutant carriers that we have done in collaboration with Roy Alkali in his lab both at Columbia and now in Tel Aviv. We know that in sporadic PD there is a reduction in the GCase index, and we also see that there is a convergence of lysosomal and inflammatory defects in GBA mutant astrocytes that could be normalized by LRRK2 kinase inhibition, and this was mouse work. So these data suggest a crosstalk between these two proteins. And so given the alteration in LRRK2 expression and GCase activity in sporadic PD, it is possible that altered enzymatic activity of these PD-related proteins in immune cells may serve as a potential biomarker, a cellular biomarker, if you will, for PD, underscoring the importance of understanding how to assay it using reliable and robust assays. So Roy mentioned this in his talk. This was a Michael J. Fox-funded global collaboration between my lab and Examco's lab to develop what we let Rebecca Wallings and Laura Hughes call the WAPA assay for optimized protocol for PBMC assessment. And the idea was, let's find a way to standardize these collection practices, figure out what is the best and most efficient way to collect these PBMCs, cryopreserve them, cryo-recover them, and see if the immune cell function is altered and in what way, so that we can then study the phenotypes of the various populations in the dish using flow cytometry and biochemistry. And the important thing was that we didn't want this to be a site or operator dependent outcome. So we did a lot of work sending samples to each other in a blinded manner. Shalini Padmanabhan, who is here from the Fox Foundation, was instrumental in motivating this collaboration. And in the end, we ended up finding that LRRK2 expression was increased in idiopathic classical monocytes, that LRRK2 enzymatic activity was also increased in idiopathic classical monocytes, and that there was increased correlation between LRRK2 levels and enzymatic activity in antigen presentation in sporadic PD classical monocytes. And finally, the decreased GCase activity, again, in classical monocytes. So we then moved on to what does it look like in patients that carry either LRRK2 G2019S or GBA1 mutations. And so again, this was a very complex design, but the Barcelona group sent us GS carriers, and Roy sent us the GBA carriers. Some went to my lab, and some went to Examco's lab, and we ended up doing panels to look at LRRK2 as well as GBA activities. This is what the study participant demographics was like. And so there is a significant difference in the age, seeing how the LRRK2 non-manifesting carriers were younger. Everything else was pretty standard and common. And so in order for you to understand the data I'm going to show you that's unpublished, I'm going to explain to you the subsets of human monocytes and what they do. So a classical monocyte is your typical CD14 high, CD16 low monocyte. They are very critical for phagocytosis. The myeloperoxide in them is very detectable and high. They generate reactive oxygen species. They're important for adhesion, migration, and antimicrobial responses. Now, and they make up about up to 90% of total monocytes in the human blood. Then there's the intermediate monocyte, which is the CD14 high, CD16 high, and these make up about 5 to 10%. And they have critical roles in antigen presentation, regulation of apoptosis, transnuclear migration, and they're high producers of IL-12 and interferon gamma. And finally, the least abundant or non-classical monocyte, they are CD14 positive, CD16 super positive, right? And they make up 5 to 10%. Now, these guys are really important for complement and FC receptor-mediated phagocytosis, migration, adhesion, antiviral responses, and high TNF production. And so we developed flow cytometry panels to see all of these cells in a mix of PBMCs by gating on their cell surface markers. We like to say this is what the monocytes are wearing. When you're doing transcriptional studies, you can see what the monocytes are thinking. And today I'm only going to show you what they're wearing. For those of you that are interested in the gating strategies, we can talk about them later, but they were pretty standard. And so here's the beginning of the data. We find in the GBA1 mutation carriers that classical and intermediate monocytes definitely display lower GCase activity upon stimulation relative to the healthy control and sporadic PD that is not rescued by LRRK2 kinase inhibition. We then looked at the GBA PD carrier in terms of what their cathepsin activity or lysosomal pancathepsin activity using a probe called BMV109. And we found that the non-classical monocytes display higher interferon gamma-induced lysosomal pancathepsin compared to the control and sporadic PD that is rescued by LRRK2 kinase inhibition. And you can see that here. We then looked at antigen presentation. And the antigen presentation capacity, much to our surprise, was not different just looking at HLA-DR and HLA-DQ MFI. The next step is to challenge them with pathogens, and I will tell you that at the end. But just by stimulating with interferon gamma, which is the major inducer of class 2, there was no difference. We then looked at the LRRK2 protein levels in these cells, and we find that the GBA PD classical monocytes displayed higher LRRK2 protein levels relative to sporadic PD or healthy controls. And finally, we wanted to see whether the LRRK2 activity was different. And so we used a phospho-Rab10 probe or antibody to look at the different monocyte subsets, and we find that GBA PD classical monocytes display higher LRRK2 kinase activity as measured by phospho-Rab10 relative to the sporadic PD and healthy control that is rescued by kinase inhibition. So to summarize the findings, we see reduction in GCase activity in stimulated GBA PD classical and intermediate monocytes, not rescued by MLI2. We see higher interferon gamma stimulated pancathepsin activity in GBA PD non-classical monocytes. We see that interferon gamma induced antigen presentation is not different from sporadic or controls. We see a higher baseline in LRRK2 levels in GBA PD classical monocytes relative to controls that is rescued by MLI2, and no difference in the interferon gamma induced LRRK2 increases from IPD or healthy control. And finally, the interferon gamma induced LRRK2 activity measured by phospho-Rab is definitely higher in GBA PD classical monocytes relative to IPD and healthy control, and is rescued by MLI2. So the next step is to also, as part of this funding, is to expose them to pathogens that maybe have been implicated in Parkinson's pathogenesis. And so once again, we're splitting up the work. The Sydney team is looking at Mycobacterium, abscessus, and tuberculosis. And we are looking at Listeria monocytogenes and Pseudomonas aeruginosa. And we've gotten some really interesting findings on the LRRK2 cohort in that the LRRK2 G2019S mutation seems to confer an immunological advantage when the patient is young or when the mice are young, but it kind of flips on you in terms of lysosomal function and antigen presentation when you're old. And so Becky Wallings, who did part of this work, has a paper in Science Translational Medicine, hopefully in final revision, showing that the mutation is conferring a type of innate immune exhaustion in monocytes, and perhaps there might be a path forward leveraging the cancer field and looking at the effect of checkpoint inhibitors. So the take-home message here is that in addition to neuroinflammation, there's increasing recognition that peripheral immune dysfunction and inflammation may contribute to PD pathogenesis. And LRRK2 and GCase are promising biomarker therapeutic targets for PD, and evidence suggests that both proteins are highly expressed in peripheral blood monocytes and are involved in similar biological pathways involving immune function and inflammation. And because there's this crosstalk, I would propose that looking at peripheral blood mononuclear cells is like having a window into the brain. And you can also look at those profiles and perhaps begin to stratify patients for clinical trials, begin to monitor the responsiveness in terms of immune effector functions in certain clinical trials longitudinally. And finally, one of my dreams is to be able to do the clinical trial in the dish first and find your responders so that you can enrich your trials for those responders whose immunophenotype you were able to reverse by your drug and easily doing, target engagement ex vivo and then obviously in vivo. So sampling PBMCs before and after. So finally, the important thing is the response to inflammatory stimuli that is altered in PD rather than the baseline levels of inflammatory markers is a much more useful and reliable cellular biomarker, because we want to be interrogating dysfunction of immune traits rather than the resting immune states of how many what cytokines the patient had that, when they came in for the blood draw. Those are heavily influenced by how much sleep you got, by how much stress you're under, by diet, by so many other things. But we believe that studying the cells that are the major producers of these cytokines and chemokines will lead us to a better understanding of how immune dysfunction is contributing to risk and progression of these diseases. And with that, I want to give credit to the team who moved with me from Emory in 2019 to the University of Florida, survived the pandemic and did quite well. Also to acknowledge our collaborators, Nick and Laura at University of Sydney, Roy and his team at Columbia and now Tel Aviv. They have an amazing and very steep learning curve for harmonization of the PBMC collection, and we're so privileged and excited to work with them in this next stage. And also to Nick McFarland at University of Florida and Ali Garrido in Barcelona for the GS patients. Thank you.
Thank you, Malu. Time for questions. Greg.
In Gaucher patients who have significant visceral organ involvement, there is clear evidence of cytokine and chemokine excesses and effects on brain function. In some patients, it resembles the sickness syndrome. And in patients that we had in Egypt in particular in 2010, when there was a worldwide enzyme shortage for treatment of Gaucher disease, we could watch mostly these type 3 patients regress in terms of their neurological function, and then when we retreated them, they became better again. And this includes things like eye movement abnormalities. So the question here with your work is, if there is both an immunologic involvement in the brain and also in the periphery, is there a place to treat the peripheral disease, maybe even in the heterozygotes, to see if it would have an effect on the brain before we get something as blood-brain barrier penetrant or with it?
Absolutely. You kind of described one of our ones that we just wrote, so maybe you'll be reviewing it too. Just kidding. So yes, some of the data that I didn't have time to show you today because it's progranulin related. One of the most upregulated genes and proteins in the progranulin knockout, as you know, is GPNMB. GPNMB increases can be detected in the brain, maybe 12 months, but when you look in the periphery, you see it at 3 months. And this is a paper that's under review at J Neuroinflammation. So I suspect that a lot of these deficits are happening earlier in the periphery, and we just haven't been looking. And I think that if we were to do that, there is a chance that we could potentially prevent the progression if there is spread or just more involvement of the brain. I don't think many of these diseases start in the brain. I think we notice them because of how disabling they are to cognition and to motor function.
Quickly, in response to that, though, patients with Gaucher disease who have been treated or untreated, both seem to be about equal risk, so that those that were treated, it doesn't prevent their Parkinsonism.
Yeah. But it's just a question of interactions that might be there.
Yeah.
Okay, so I'm getting the signal here that everyone is hungry.
Lunch.
So maybe over lunch we can continue the questions, and thank you everyone and thanks the speakers.